Okay, so hi everyone, welcome to my channel. Today I'm going to give you a brief knowledge explanation about mitosis. So before we start, I want to ask you guys a question. Have you ever noticed some certain phenomenon where even if you don't turn to treat your wounds after getting hurt, you will finally observe some significant signs of healing after several days? Or have you ever looked in the mirror and noticed that you appear larger than before? So you might wonder what nature process contributes to the regeneration of skin and tissue. So all of this process is related to our today's topic, mitosis. Here is a brief introduction of mitosis. Mitosis is a significant circular process essential for growth, development, maintenance of organisms. It involves the division of one single parent cell into two genetically identical daughter cells, each containing a complete set of chromosomes. Mitosis consists of four distinct stages: prophase, metaphase, anaphase, and telophase, where the chromatin condenses into visible chromosomes, aligns at the metaphase plate, and separate into sister chromatins, and eventually years into two daughter cells. So, after my brief explanation, do you want to explore more about the mitosis process and have a better observation under the microscope? Here is a stimulated experiment. Here are some materials that we need to use in this experiment. Three types of solution: faceative solution, Jensen-Wallace solution, hydrochloric acid, and the most important one, microscope. And we also need to use a garlic root tips as our material, pencil, scissors, four cups, and microscope slides and cover slips. Here is our steps. Firstly, we use a pair of scissors to cut several 1 centimeter sections of actively growing garlic root tips and place them into a glass container filled with fixative solution to ensure the root tips are fully submerged, and we allow the root tips to remain undisturbed in the fixative solution for 3 minutes. And second step, we use the 4 caps to transfer 4 or 5 root tips to the glass watch. We add 4 to 5 drops of the hydrochloric acid to the glass watch to cover the root tips completely, and we need to allow the root tips to soak in the acid solution for 5 minutes. Then we gently use water to wash the root tips with 2 to 3 times to remove the acid solution. And the next step, we apply 2 to 3 drops of 0.8 Jensen-Wallace stain to cover the root tips and ensuring they are fully immersed. And we should allow the root tips to soak in the solution for 3 minutes. And the similar steps, we still need to wash the root tips with water, repeating the process 2 to 3 times. And then we place a single root tips on a clean glass slide and add 1 drop of water to the root tips. We use the eraser of end of the pencil to gently press down to squash the root tissue. We apply sufficient pressure to obtain the single layered cells. We need to take care that we not break the cover slips or the cell wall. And then finally, we can start our observation. So, for our observation, under the microscope, we can successfully observe all 4 stages of mitosis. The first one, prophase, as we can see in both the video and picture, prophase initiates the mitosis. It's condensing the chromatin into visible chromosomes, and it's disseminating the nuclear envelope and forming the mitotic spindle. It sets the stage for accurate chromosome segregation and circular division. The second stage, metaphase. Metaphase is the second stage of mitosis. It follows prophase and precedes anaphase. In the metaphase, condensed chromosomes align along the metaphase plates, And the metaphase serves as a critical checkpoint, ensuring that each daughter cell receives the correct number of chromosomes. The third phrase, anaphrase, it follows the metaphase and precedes the telophase. During the anaphrase, sister chromatins previously aligned the metaphase plates, separate and move towards the opposite points of the cell. This movement is facilitated by the shortening of microtubules attached to the centromeres, ensuring the equal distribution of genetic material to the daughter cells. The last one, telophase. Telophase is the final stage of mitosis. It follows the anaphase. During the telophase, the separated sister chromatins arrive at the opposite points of the cell and decondense back into chromatin. Nuclear envelopes begin to form around each set of chromosomes, establishing distinct nuclei within the cell. Additionally, mitotic spindle fibers disassemble, marking the conclusion of chromosome movement. After the explanation about the fourth stage of mitosis, and here I also want to add some application of mitosis, not only in the daily life we mentioned at before, but also in some research field. Mitosis can be applied in the agriculture crop improvement. It is essential for some plants through the methods like cutting and tissue culture. It allows for the rapid multiplication of desirable plants and the production of genetically identical clones with desirable traits, aiding in the crop improvement and agriculture practice. Secondly, it can help in some drug development. Its targeting the mitotic process has led to the development of anti-cancer drugs, and which can disrupt mitotic spindle function and inhibit the cell division in cancer cell. Lastly, mitosis also plays a central role in the development generated through IVF and other assisted reproductive technology. This technology also uses the mitosis. So here we approach the end of our video. So before the end of our video, I want to thank most is my Professor Lee, the professor of my bio class. Thank you for your insightful lectures, challenging assignment, and encouragement. Besides, I also want to thank my ThinkPairShare group member. Thank you guys, excellent ideas, and our group's cooperation. So thanks for your watching.